Understanding the Western blot technique. Western blotting, also known as immunoblotting, is a widely used analytical technique in molecular biology and biochemistry to detect and quantify specific proteins within a complex mixture. First developed in the late 1970s, it remains an essential tool for protein analysis. The process begins with protein extraction from a biological sample, such as cells or tissues. Proteins are isolated using lysis buffers that contain detergents, salts, and protease inhibitors to ensure protein integrity. The concentration of extracted proteins is often quantified using assays like the Bradford or BCA assay. Next, proteins are separated by SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS, a detergent, denatures proteins and imparts a uniform negative charge, allowing them to migrate through the gel based on molecular weight. Smaller proteins travel faster, while larger proteins migrate more slowly. After electrophoresis, the separated proteins are transferred onto a nitrocellulose or PVDF membrane. This transfer can be performed using either a wet or semi-dry electroblotting system. The membrane immobilizes the proteins, making them accessible for antibody-based detection. To reduce background noise, the membrane is blocked with a solution, typically containing milk or BSA, bovine serum albumin. Blocking prevents non-specific binding of antibodies to the membrane. The detection phase involves the use of primary and secondary antibodies. The primary antibody specifically binds to the target protein. A secondary antibody, conjugated to an enzyme like horseradish peroxidase, HRP, or alkaline phosphatase, AP, recognizes and binds to the primary antibody. These enzyme-conjugated secondary antibodies amplify the signal, enhancing detection sensitivity. To visualize the protein, a substrate is added that reacts with the enzyme on the secondary antibody. This reaction generates a detectable signal, often chemiluminescent or chromogenic. Chemiluminescence is widely used, and the signal is captured on X-ray film or with imaging systems. Modern methods also use fluorescently labeled antibodies, which allow for simultaneous detection of multiple proteins. Western blotting has a broad range of applications, including 1. Protein expression analysis. Measuring protein levels across different conditions, such as cell treatments or disease states. 2. Post-translational modification studies. Detecting modifications like phosphorylation, glycosylation, or ubiquitination. 3. Validation of other techniques. Confirming results from proteomics or RNA-based studies. 4. Disease diagnostics. Identifying biomarkers or confirming infections, such as HIV diagnosis. While Western blotting is highly specific and versatile, it has limitations. It requires antibodies that are highly specific to the target protein, and optimization of experimental conditions can be time-consuming. Additionally, the technique has limited quantitative capability compared to newer methods like mass spectrometry. Advancements in Western blotting have enhanced its efficiency and sensitivity. These include automated systems, improved transfer techniques, and more robust detection reagents. Quantitative Western blotting with fluorescent dyes or digital imaging has further expanded its utility in modern research. In summary, Western blotting remains a cornerstone technique in molecular biology. By enabling precise detection of specific proteins, it provides critical insights into protein expression, modification, and function.